So if you happen to be taking like a statin for your cholesterol or maybe a calcium channel blocker for your blood pressure.
Right, common medications.
Yeah, super common. Your doctor has probably given you a very specific and honestly somewhat bizarre instruction. They tell you that under no circumstances should you drink grapefruit juice.
Yes, the classic grapefruit warning.
Exactly.
Right.
But here is where it gets crazy.
Mm.
If you were to take a wild yellow mushroom, specifically one pulled from the damp, decaying leaf litter of a European hardwood forest, and you somehow introduce its microscopic root network to that grapefruit juice.
Just hypothetical kitchen chemistry here.
Right. If you did that, the chemical threat to your medication would be neutralized by up to 60%.
Oh, wow.
Yeah, up to 60%. And the thing is, this mushroom has absolutely no concept of human pharmacology. Doesn't know what a prescription drug is.
I mean, why would it?
Exactly. Yet it possesses this highly evolved biochemical shield that is perfectly designed to dismantle the exact compound in grapefruit that causes accidental overdoses in humans.
It is a stunning biochemical coincidence, and honestly, when you really look at this organism, it is perhaps the least bizarre thing it does to survive.
Wait, seriously? Fixing our medication problem is the least bizarre thing.
Without a doubt, when you strip away the culinary prestige surrounding this mushroom, you are left with a biological entity that actively farms other species.
Farms. Like agriculture.
Literally agriculture. It also deploys volatile rocket fuel components as a defense mechanism, and it uses a shape-shifting survival strategy that makes it one of the most evolutionarily sophisticated fungi we have ever mapped.
Okay, that is exactly why we are doing a deep dive into this today. We are pulling apart the 2024 to 2026 genomic profiles, agricultural field studies, and biochemical analyses on Morchella esculenta.
The famous yellow morel.
The yellow morel, exactly. And our mission today is to completely bypass the culinary stuff. No butter and garlic recipes here.
No, we are looking at the actual ecology.
Right. Because if you were to walk through an old apple orchard and just step on the soil where one of these is growing, you'd be compressing thousands of microscopic highways designed for bacterial transport.
It's an invisible infrastructure.
It really is. We want to look under the hood of an organism that has spent the last 100 million years perfecting this highly aggressive form of subterranean engineering.
But to really grasp the scale of that engineering, we first have to look at the massive blind spot we've had regarding its identity.
Oh, right, the taxonomy issue.
Yeah, for centuries, mycology operated under a fundamental assumption about Morchella esculenta that recent molecular phylogenetics has proven entirely false. Biologists are now calling it a 100-million-year identity crisis.
An identity crisis that goes all the way back to the foundation of taxonomy itself, right?
Yeah.
Like Carl Linnaeus in 1753.
Exactly. Linnaeus is looking at this honeycomb-capped mushroom and, relying entirely on its physical morphology. He names it Phallus esculentus.
Which is based on a rather obvious visual similarity to other things.
Right, a classification based on visual shape alone.
Yeah.
Christian Hendrik Cursoon thankfully corrected that in 1801.
Thank goodness for that.
He reclassified it as Morchella esculenta. But the underlying error in their methodology actually persisted for another 200 years.
Because they were still just classifying it based on what it looks like.
Exactly. It was purely morphological. Scientists and foragers globally just assumed that if you found a yellow morel with that distinctive pitted cap in a forest in France,
Yeah.
and you found an identical-looking yellow morel in the woods of Pennsylvania, you were dealing with the exact same globally distributed species.
Which, I mean, is a totally fair assumption to make. If two complex structures look identical, even down to the microscopic level, your intuition says they have to be the same organism.
It makes sense intuitively, sure.
But when you look at the 2024 to 2026 genomic sequencing, specifically when researchers started amplifying the internal transcribed spacer, or ITS regions of their DNA, that illusion shatters completely.
It completely falls apart. The genomic data revealed that the true Morchella esculenta exhibits high continental endemism.
Meaning it's geographically isolated?
Exactly. It is almost entirely confined to Europe and Asia. So those yellow morels that foragers have been pulling out of North American forests for centuries.
The ones everyone called Morchella esculenta.
Right. They're actually entirely distinct species. They've now been separated into taxa like Morchella americana or Morchella esculentoids.
It's basically like discovering that your lifelong best friend actually has an identical twin you've never met, and you've been hanging out with both of them interchangeably for years without realizing it.
That is a perfect analogy. It is a textbook example of convergent evolution.
It reminds me of the aerodynamic design of high-speed bullet trains. Like, if you have engineers in Japan and engineers in France trying to build the fastest train possible.
They both end up with that same elongated nose.
Right. The same sleek profile. Not because they stole each other's blueprints, but because they are solving the exact same physics problem, air resistance.
Yes. These mushrooms face similar environmental pressures in North American and Eurasian temperate forests.
So they converged on the exact same physical architecture, even though their internal genetic wiring diverged eons ago.
That's a highly accurate way to frame it, especially when you look at the timeline of that divergence.
How far back are we talking?
Well, when they map the mutation rates in their DNA to establish a molecular clock, the data is wild. It shows that the Esculenta clade, the yellow Morels, and their distant cousins, the Elata clade.
Those are the black Morels, right?
Correct. They diverged from their last common ancestor roughly 100 million years ago.
Wow. So we are talking about the late Cretaceous period here.
Exactly. The continents are still shifting out of the Pangaean supercontinent phase.
The climate is wildly different. You literally have apex dinosaur predators dominating the terrestrial ecosystems.
And that is when this specific genetic lineage branches off, which introduces a really fascinating concept called phylogenetic niche conservation, or PNC.
Because what's truly staggering isn't how much it changed over 100 million years, but how much it didn't change at all, right?
Precisely. Despite surviving the KT extinction event that wiped out the dinosaurs.
And multiple ice ages.
Right, and massive shifts in global flora. Despite all of that, Morchella esculenta remained hyper-specialized. It exhibits an intense evolutionary stubbornness.
It knows what it likes, and it refuses to budge.
Exactly. It demands temperate, deciduous biomes. It insists on highly specific soil chemistry, specifically alkaline limestone-heavy substrates.
And it only associates with hardwoods.
Yes. Ash, elm, sycamore, and famously old apple orchards.
Which is such a stark contrast to those black Morels of the Elata clade.
Yeah.
They took the complete opposite evolutionary route.
They did. They are the generalists of the family.
They tolerate acidic coniferous forests, they handle high altitudes, and they are notorious for fruiting in these massive flushes right after a forest fire.
They love disturbed ground. They thrive on that sudden influx of carbon from a burn.
But the yellow Morel, our Morchella esculenta, avoids that entirely. It wants the stable, slowly decaying leaf litter of a lowland floodplain.
It is a highly specialized niche. Which brings us back to that physical architecture you mentioned earlier, the bullet train design.
The honeycomb cap. It's so iconic.
It is one of the most recognizable structures in nature. But when you look at the biological breakdown in these recent papers, you realize it isn't just for looks.
No, it's actually an intricate aerodynamic mechanism.
Let's break down the structure of that cap. You have the pits, the deep indented areas forming the honeycomb, and you have the raised ridges that form the outer net-like pattern.
And the sources point out something fascinating here. Only the pits are fertile.
Right. They're heavily lined with acini, which are the microscopic sacs that forcibly eject the spores.
But the raised ridges, the parts that stick out the furthest, are completely sterile. They do not produce a single spore.
Not one.
Which, honestly, from a purely biological standpoint, seems crazy. If your singular evolutionary goal is reproduction, leaving the most exposed surface area completely sterile feels like a massive waste of biological real estate.
It does seem counterintuitive. Why not coat the entire structure in acid to maximize spore output?
Exactly. Just blast spores from everywhere.
Well, because more spores don't equate to better dispersal if the fluid dynamics of the environment are working against you.
Okay, so how does the environment work against them?
If the raised ridges produce spores, those spores would be ejected directly into the turbulent boundary layer of air moving immediately over the mushroom's surface.
So they just get caught in the turbulence.
Worse, they would collide with spores being ejected from neighboring ridges. You'd essentially create a microscopic traffic jam.
Oh, wow.
Yeah, the spores would lose all their kinetic energy, they'd clump together due to electrostatic forces, and they'd fall directly to the ground right at the base of the parent mushroom.
Which defeats the whole purpose. They'd just be competing with themselves for the exact same depleted resources.
Exactly. So it actively engineers the airflow instead. By leaving the ridges sterile, it creates dedicated physical wind channels.
So the fertile acini are recessed safely inside those pits.
Right. When they build up enough turgor pressure to rupture, they shoot their spores outward.
They clear the physical barrier of the sterile ridges.
And they enter the laminar microcurrents of air that are flowing cleanly over the honeycomb structure.
The ridges are basically acting as aerodynamic stabilizers.
Exactly like stabilizers. They guide the air over the pits so the spores are caught in an optimal updraft, maximizing wind dispersal.
It is a structural adaptation perfected over 100 million years of trial and error, just sitting there in the dense, stagnant air of a forest floor, figuring out aerodynamics.
This is beautiful engineering.
Okay, so we are looking at an organism that is aerodynamically optimized, intensely stubborn about its soil pH, and has maintained this strict specialization since the Cretaceous. On paper, that sounds incredibly fragile. Like, usually, hyper-specialized organisms are the first ones to go extinct when an environment shifts, simply because they can't adapt.
That is the standard ecological rule, yes.
But when you look beneath the soil matrix, the mycelial network of Morchella esculenta is doing something entirely different. It survives because it employs this shape-shifting trophic strategy.
The term the literature uses is facultative trophic strategy, and it completely upends our traditional categorization of fungal roles in a forest ecosystem.
Because we usually put fungi into very rigid metabolic lanes.
We do.
Right.
Generally, you have your saprobes, which are the decomposers.
They just eat dead stuff.
Right, they secrete extracellular enzymes like lignin peroxidases and cellulises to break down dead organic matter fallen timber, dead leaves. They are the chemical recyclers of the forests.
And then on the other side, you have the mycorrhizal fungi.
Exactly. The mycorrhizal fungi form these mutualistic, symbiotic relationships with living plants.
So they physically connect to the trees.
Yes. Their hyphae wrap around or literally physically penetrate the root tips of a tree. They act as an incredibly fine, highly efficient extension of the tree's root system.
Scavenging for water and trace minerals from micropores in the soil that the tree roots are just too big to reach.
Exactly. And in exchange for delivering those minerals, the living tree photosynthesizes and pumps complex carbohydrates, liquid sugars, down into its roots to feed the fungus.
It's a trade deal.
A very stable trade deal. And most fungi are metabolically locked into one of those two lanes. A decomposer can't suddenly become a symbiotic partner.
They just don't have the genetic machinery to make the switch.
Right. But the sequencing shows that Morchella esculenta operates on a sliding continuum. It can seamlessly shift its entire metabolic profile.
It's like a freelance worker who can effortlessly switch from being an independent contractor like a saprobe to a full-time corporate employee, a symbiont, whenever the economy demands it.
That's a great way to think about it. If it's growing near a healthy ash tree, it forms those mycorrhizal associations, trading soil minerals for a steady stream of carbon from the tree.
But if that tree gets diseased, or the environment shifts and the carbon flow stops, the morel doesn't starve.
No, it instantly upregulates a completely different set of genes, switches its enzyme production, and becomes an aggressive saprobe. It just starts scavenging and decaying the dead plant matter around it.
And it is super aggressive when it's in that state. Several recent papers characterize its mycelium as a mineral miner.
Yes, it doesn't just passively absorb nutrients that happen to wash over it in the groundwater.
It goes looking for them.
Actively. The hyphal tips seek out resistant organic compounds and specific mineral deposits in the soil matrix, and they secrete targeted acids to dissolve and extract them.
And it's mining those minerals for a very specific purpose, right? Stockpiling.
It has to stockpile. Because the environment of the forest floor, the temperature swings, the moisture levels, is inherently volatile.
So to survive that volatility, without the permanent protection of a living tree root, it builds an underground bunker.
The sclerotium, this is perhaps its most crucial survival mechanism.
How does that actually work? What triggers it to build a bunker?
When the mycelial network senses incoming environmental stress, maybe the soil moisture drops indicating a drought, or temperatures begin a sustained decline toward winter freezing, the network completely stops its outward vegetative expansion.
It just halts in its tracks.
It halts, and the hyphae begin to tightly aggregate. They repeatedly branch and weave into these dense, hardened subterranean masses.
Structurally, how is the sclerotium different from just like a clump of mycelium?
It undergoes a profound biochemical transformation. The outer layers of the hyphae and the sclerotium undergo heavy melanization.
Melanin, like what's in human skin.
The exact same pigment. In fungi, melanin acts as an incredibly durable biological shield. It protects the inner cells from UV radiation, enzymatic attacks from soil predators, and extreme temperature fluctuations.
That's amazing.
And inside this melanized rind, The cells pack themselves with massive droplets of lipids and glycogen.
They're essentially nutrient batteries.
Precisely. These structures are usually no larger than a walnut, completely invisible beneath the soil, and they can lie dormant for months or even years, just weathering catastrophic surface conditions.
And then eventually, they act as the energy reservoir for the fruiting body.
Yeah.
But the timing of when they choose to fruit is what really caught my attention in the sources.
Ah, the thermal window.
Yeah. Morchella esculenta doesn't just wait for a nice, warm summer day to utilize all that stored energy. It deliberately activates when the soil is still brutally cold.
It's a strategy of temporal evasion. It has evolved specialized cold-adapted enzymes that allow its metabolic processes to kickstart at incredibly low soil temperatures.
We're talking just a few degrees above freezing, right?
Yes, long before the spring canopy has even begun to leaf out.
From a competitive standpoint, the logic there is just brilliant.
How so?
Well, the soil is a fiercely contested microbial battleground. If you wait until the soil hits 15 or 20 degrees Celsius, you are waking up at the exact same time as millions of other fungal species.
Right, and bacteria and nematodes.
All of them violently competing for the exact same finite pool of free carbon and nitrogen.
Exactly. By evolving to germinate and fruit in that narrow, near-freezing thermal window, Morchella esculenta effectively sidesteps the competition entirely.
It establishes its network and begins pushing up its massive fruiting bodies when the rest of the soil microbiome is still formally suppressed. It's basically beating everyone else to the punch.
It monopolizes the resources through sheer metabolic precocity. However, just escaping the competition isn't enough to sustain the massive energetic cost of producing those fruiting bodies.
Especially in soils that might be depleted of easily accessible nutrients.
Right. And this is where the 2024 to 2026 data truly shifts our fundamental understanding of fungal ecology.
This is the mind-blowing part.
Morchella esculenta isn't just reacting to its environment or scavenging. It is actively engineering its own microbial food supply. It is an agriculturist.
Fungal agriculture. I remember reading that term in the papers and thinking it had to be a metaphor.
It is not a metaphor.
We associate agriculture with a high degree of cognitive or social complexity. Humans farm. Leafcutter ants farm fungi. Certain termite colonies manage fungal gardens.
Organisms with complex societies.
Exactly. But to say a fungus, an organism with no central nervous system, no brain, literally just a distributed network of filamentous tubes, is actively farming a specific bacterium.
It forces a complete reevaluation of what constitutes complex behavior.
Let's break down the mechanics of this, because the three-phase agricultural cycle described in the sources is staggeringly precise. It specifically farms a soil bacterium called Pseudomonas putida.
Yes.
Why is phase 1 called the fungal highway?
Well, to understand phase 1, you really have to look at the microphysics of soil. Soil is not a solid block.
Right, it's mostly dirt and rocks.
It is a porous matrix of mineral particles, sand, silt, clay, and they are separated by microscopic gaps. Many of those gaps are actually filled with air.
Okay.
Now, a bacterium like Pseudomonas putida is flagellated, meaning it swims.
Has a little tail.
Exactly. It requires a continuous film of water to move. So when it encounters an air-filled pore in the soil, it hits a physical dead end. It cannot cross the gap.
So the bacteria are essentially trapped in isolated microscopic puddles. Their mobility is severely restricted.
Precisely. They can't forage for food effectively. But the hyphae of Morchella esculenta are physical structures extending directly through those air gaps.
Like bridges.
Yes. And crucially, because the fungus is respiring and moving water through its network, the surface of its hyphae is coated in a continuous microscopic film of water.
Oh, wow. So the bacteria use it.
They absolutely do. The Pseudomonas putida bacteria migrate onto the fungal network and use it as a physical superhighway. They swim along the water film on the outside of the hyphae.
That allows them to rapidly bypass all the air gaps and colonize vast new territories in the soil at speeds they could never achieve on their own.
It builds the transit infrastructure for them.
But it doesn't just passively allow them to hitch a ride. That brings us to phase 2, rearing. The fungus intentionally feeds the bacteria once they're on the highway.
It does.
But wait, farming implies an anticipation of a future return on investment. How does a blind network of tubes know to divert its hard-earned carbon resources, the energy it needs for its own sclerotia, to feed a separate organism?
It is important to realize it's not a cognitive decision. It is a highly evolved, localized biochemical feedback loop.
Okay, how does the loop work?
When the hyphae detects the physical presence and the specific chemical signatures of Pseudomonas putida on their surface, it triggers a localized metabolic shift.
So the fungus senses them.
Yes, and the fungal cells in that specific area begin to exude carbon-rich compounds, essentially liquid sugars, directly through their cell walls into that surrounding water film.
Setting up free buffets along the highway.
That's a great way to picture it. They create localized, nutrient-dense nursery zones. The fungus is actively provisioning the bacteria, creating an environment where the bacterial population can exponentially replicate.
So it builds the highway, it pumps free resources into specific zones to artificially inflate the bacterial population. It's quite literally cultivating a crop.
It is. Which brings us to phase 3, harvesting.
The dark turn. Because once that bacterial population reaches a critical density, the biological generosity completely stops.
The dynamic completely inverts. The localized exudation of carbon ceases. The fungus switches its enzymatic output and begins to secrete enlytic enzymes.
Which would do what,
They
exactly?
specifically target the peptidoglycan cell walls of the bacteria. It breaks them down, effectively digesting the crop of Pseudomonas putida it just spent significant energy cultivating.
It just eats the commuters on the highway.
It does. It absorbs the resulting bacterial lysomate, the nitrogen, the carbon, the trace minerals, and translocates those nutrients back through its network to fuel its own growth.
Primarily to fill those subterranean sclerotia we talked about.
Exactly.
Okay, but here is my big question. Out of the millions of bacterial species in the soil microbiome, why go through these incredibly complex, energy-intensive, three-phase operations specifically for Pseudomonas putida? There must be easier sources of carbon in the soil.
There absolutely are easier sources of carbon, and that is the absolute crux of this discovery. Morchella esculenta isn't farming Pseudomonas putida just for calories.
What is it farming them for, them?
It is farming them for chemical remediation. It is using the bacteria as a biological scrubber to survive its own metabolic exhaust.
That sounds like science fiction.
It really does, but it all hinges on a compound called ACC.
Right, one aminocyclopropane, one carboxylic acid. I was looking at the chemical breakdown of the soil environment and the sources, and there's a massive anomaly here.
Yes, walk us through the anomaly.
As a fungus grows in a densely packed, continuous environment, like a subterranean soil matrix, its natural respiration and metabolic processes produce ACC. And ACC acts as a direct precursor to ethylene gas. In agricultural science, we usually think of ethylene as the plant hormone that ripens fruit, right? Like putting a banana in a paper bag.
Yes, that's the most common context.
But in fungal ecology, a buildup of ethylene in a closed soil system is highly toxic. It acts as a powerful stress hormone.
It is incredibly detrimental to the fungus.
It arrests hyphal extension, it induces premature aging in the cells, and it effectively shuts down fungal growth completely. By all logic, the soil should be choked with ethylene gas from the Morels' own growth, and it should just kill itself.
That it doesn't.
Because of the Pseudomonas putida.
Exactly. This specific bacterium possesses an enzyme called ACC. Nitrogen, of course.
So they eat the poison before it becomes poison.
Right. They break the ACC down before it can ever convert into ethylene gas.
Wow.
By intentionally cultivating massive populations of Pseudomonas putida along its network, the Morel is actively deploying a localized filtration system.
The bacteria eat the toxic ACC precursor, keeping the localized ethylene levels artificially low.
Which is what allows the Morchella esculenta network to grow significantly larger, denser, and longer than its physiological limits would naturally allow.
It is environmental engineering on a microscopic scale. It's not just surviving its ecosystem, it's actively manipulating the chemical atmosphere of its habitat to prevent its own suffocation.
It is mastering its environment.
And that level of biochemical manipulation naturally extends to how the mushroom deals with the external threats in its environment, too.
Yes, it has to defend itself.
If the soil is packed with the degrading plant matter, especially those specific hardwoods and old apple orchards it prefers, that soil is essentially a chemical minefield of plant defense compounds.
It is a highly toxic soup down there. Plants do not want to be eaten, and they certainly do not want to be degraded by fungi.
So they fight back.
They do. They defend themselves by secreting potent secondary metabolites into their leaves and roots. When those leaves fall and decay, all those toxins leach directly into the soil.
So to survive as a sap probe, as a decomposer, in that specific decaying leaf litter, Morchella esculenta had to evolve an incredibly robust chemical shield.
It had to become a master chemist.
Which brings us directly back to our opening hook. the grapefruit hack. The intersection of this fungal defense mechanism and human pharmacology. Break down the specific plant toxin we are dealing with here.
The compounds in question are a class of organic chemicals called furanocoumarins, specifically things like sorrelin or bergamotin.
And plants produce a lot of these.
Many plants do, especially citrus trees like grapefruit. If an insect eats a leaf containing furanocoumarins, or a fungus tries to digest it, the chemicals profoundly disrupt their cellular function.
But as we discussed earlier, if a human consumes these furanocoumarins, say, by drinking a glass of grapefruit juice, they pass into our digestive tract.
And they specifically target an enzyme in our liver and small intestine known as CYP3A4.
Which is basically the chemical incinerator for our prescription drugs, right?
Right. That's a good way to put it. Its primary job is to metabolize foreign compounds in the bloodstream, breaking down medications so they can be safely excreted by the body.
So what happens when the grapefruit juice hits that enzyme?
When furanocoumarins hit the CYP3A4 enzyme, they act as what biochemists call a suicide inhibitor.
That sounds aggressive.
It is. They bind irreversibly to the enzyme, permanently knocking it offline. Until your body can synthesize a completely new batch of that enzyme, which can take hours or even days, any medication you take is not being metabolized.
Whether it's a statin or a blood pressure pill.
Exactly. The drug just continues to circulate and accumulate in your bloodstream, quickly reaching toxic, potentially fatal concentrations.
But the 2024 to 2026 profiling of Morchella esculenta shows that its mycelium has a direct countermeasure for this exact compound.
It does. Researchers discovered that the cell walls of the Morchella esculenta mycelium are studded with specific binding sites, and these sites possess an extraordinarily high affinity for furanocoumarins.
So when the fungal network encounters these plant toxins in the soil,
The binding sites act like chemical magnets. They latch onto the furanocoumarins and physically pull them out of the surrounding solution, effectively neutralizing them before they can penetrate the fungal cell and cause any damage.
and because biochemistry basically operates purely on molecular shapes, those fungal binding sites don't care if the furanocoumarin came from a rotting apple leaf in the soil or a glass of Florida grapefruit juice.
The molecular key fits the lock perfectly.
It's wild. The studies show that introducing the mycelial extract to grapefruit juice bound and neutralized up to 60% of the furanocoumarins.
Drastically reducing their ability to inhibit the human CYP3A4 enzyme. We are looking at potential applications where we could use this fungal binding mechanism to create biofilters for fruit juices.
Removing the danger for cardiac patients entirely.
Exactly. But it is vital to remember the evolutionary context here. The mushroom did not evolve this mechanism to solve human medical crises.
No, it evolved it just to survive the toxic environment of the forest floor.
Right. The pharmacological benefit to humans is just a coincidental byproduct of its sheer chemical complexity.
And that chemical complexity carries over into other active compounds discovered in the mushroom, too. The sources go into heavy detail regarding a recently isolated protein called MEP5.
Yes, MEP5.
And this isn't just a trace element. It appears to be a highly active, targeted medical compound hidden right inside the fruiting body. What exactly is it?
MEP5 stands for Morchella esculenta protein 5. Biochemically, it is a 33.12 kilodalton lectin.
What's a lectin, for those of us who aren't biochemists?
Lectins are carbohydrate-binding proteins. You can think of them as highly specific biological Velcro. They are designed to recognize and attach to very specific sugar structures on the surfaces of cells.
And MEP5 is looking for a specific sugar.
Yes, it is uniquely fucose-specific, meaning it actively seeks out and binds to fucose residues.
And the research indicates this specific fucose-binding action has profound effects on the human liver, specifically concerning NAFLD or non-alcoholic fatty liver disease.
Which is a massive global health issue.
Right. It's where metabolic dysfunction causes lipid droplets fat to accumulate inside liver cells.
Yeah.
And that leads to severe inflammation, oxidative stress, and eventually cirrhosis, completely independent of alcohol consumption.
A very severe condition.
So how does a mushroom lectin interact with human liver pathology?
The clinical studies from 2025 demonstrate that MEP5 possesses potent hepatoprotective properties.
Liver protecting.
Yes. When administered in models of NAFLD, it significantly normalized lipid profiles. It actively reduced the fat accumulation in the hepatocytes.
How is it doing that?
It achieves this by modulating the MAPK signaling pathway.
The mitogen-activated protein kinase pathway. If we look at basic cellular biology, this is essentially an intracellular relay race, right?
That's an excellent way to describe it.
A signal hits a receptor on the outside of the liver cell,
Yeah.
and a series of kinase proteins start passing a phosphate group down the chain, like passing a baton.
Moving it deeper into the cell.
Right, until the signal reaches the nucleus and tells the DNA to change its gene expression. And usually, in the case of NAFLD, it's instructing the cell to pump out inflammatory cytokines.
Precisely. The MAPK cascade, which includes proteins like ERK, JNK, and P38, is hyperactivated in a fatty liver. It drives a chronic state of severe inflammation.
So where does MEP5 come in?
The MEP5 lectin from the Morel binds to specific surface receptors and effectively downregulates that cascade. It acts as a cellular dampener.
It intercepts the relay race.
Exactly. It reduces the phosphorylation of those key proteins, which significantly lowers the oxidative stress and inflammatory response in the liver tissue.
That is incredible. So you have a wild organism that acts as a superfood. The nutritional profiles in the sources show it's dense with vitamin D, B2, B3, copper, zinc.
A very rich nutritional profile.
And it's carrying heavily active hepatoprotective lectins, plus the ability to neutralize for coumarins. It sounds like a miraculous pharmacological package.
It does sound that way, yes.
Yes. But any deep dive into the chemistry of Morchella esculenta has to come with a massive underlined warning label.
Absolutely. This cannot be stressed enough.
Because the very same chemical arsenal it uses to defend itself in the soil can be incredibly dangerous to humans.
It is a critical dichotomy. Morchella esculenta is highly toxic when raw.
Highly toxic.
And the mechanism of that toxicity is severe. The raw mushroom tissue contains significant levels of hydrazine derivatives, primarily a compound closely related to gyrometrin.
Hydrazine. Just to give that chemical some context, hydrazine compounds are literally used as high-energy propellants in rocket fuel.
They are. We are talking about volatile, highly reactive industrial chemistry.
And when you ingest these raw compounds, what happens?
The acidic environment of the human stomach hydrolyzes them. It converts them into monomethylhydrazine, or MMH.
Which is bad.
Very bad. MMH is a severe central nervous system toxin. Once it absorbs into the bloodstream and crosses the blood-brain barrier, it acts as a competitive inhibitor of pyridoxal 5-phosphate.
Which is the active form of vitamin B6.
Right? Exactly. And vitamin B6 is necessary for the brain to synthesize GABA.
GABA is the primary inhibitory neurotransmitter. It's basically the chemical that keeps your neural firing in check. It's the brakes for your brain.
Precisely. By blocking that synthesis, the MMH severely depletes the brain's GABA levels. The neurological brakes are effectively cut.
So the neurons just start firing out of control.
Yes, patients who have consumed raw or undercooked morels experience severe gastrointestinal distress, intense vertigo, visual disturbances, and ataxia.
Ataxia meaning a total loss of voluntary muscle coordination.
Yes. And in severe cases, the unchecked neural firing leads to intense seizures and can absolutely be fatal.
The clinical reports mentioned in the background of our sources actually outline several recent severe poisonings at high-end restaurants.
Sadly, yes.
Chefs attempted to serve morels lightly seared or even shaved raw over dishes to preserve their delicate texture. The results were disastrous.
Because they fundamentally misunderstood the chemistry. The crucial factor with hydrazine toxins is that they are highly volatile and thermally unstable.
They need heat.
They require sustained high heat to break their chemical bonds and vaporize completely. A quick sear is just not enough.
The mushroom must be thoroughly and completely cooked.
Yes. Once subjected to sustained heat, the hydrazine derivatives break down completely, rendering the mushroom perfectly safe and pharmacologically beneficial.
But that raw toxicity is just a stark reminder. This organism's complex chemistry evolved over 100 million years for its own survival and defense, not for our culinary convenience.
It is a fortress. It truly is.
It operates on a specific thermal window, builds heavily armored subterranean sclerotia, engineers its soil pH by farming Pseudomonas, and defends itself with volatile neurotoxins.
All highly calibrated evolutionary mechanisms.
And all of these specific mechanisms combine to explain why, for over a century, this mushroom completely defied human attempts to farm it.
You couldn't just scatter spores in a field and expect a harvest.
Right. Which leads us perfectly into the massive economic implications and the modern agronomic breakthroughs. Let's talk about the 40,000 rupee prize.
The economics surrounding Morchella esculenta are driven entirely by its elusiveness.
Because it's so hard to find.
Exactly. In the high altitudes of the Himalayas, specifically in regions of Kashmir, the dried mushroom is known locally as Gucci. And because it could not be farmed, 100% of the global supply had to be hand-foraged.
which drove the market price to astronomical heights. The sources mention it routinely selling for between 15,000 and 40,000 rupees per kilogram.
It became one of the most highly prized culinary ingredients on the planet.
But the human cost of acquiring it was immense. We're talking about local gatherers having to navigate the treacherous topography of the Pir Panjal range.
During the volatile spring thaw, no less.
Right, they are trekking through steep, freezing, avalanche-prone forests. And because of the mushroom's strict phylogenetic niche conservation, its absolute refusal to fruit unless the soil chemistry, the microbial balance, and that exact near-freezing thermal window align perfectly, a gatherer could risk their life for a week in the mountains and return with absolutely nothing.
The supply chain was incredibly dangerous and completely unpredictable, which naturally ignited a century-long global agronomic race to crack the cultivation code.
Let's trace the history of those attempts, because it's basically a long timeline of scientific frustration punctuated by localized flukes and massive misidentifications.
The classic example is Rapin in 1901. He reports to the French mycological community that he has successfully cultivated morels in a cave system.
But when you look at his methodology, it wasn't a controlled agricultural process at all.
Not even close. He had inoculated pots of soil with spores, abandoned them in the cave, and literally 9 years later, they miraculously fruited.
He couldn't replicate it, he couldn't scale it. It was a complete biological anomaly.
Yeah.
The environmental variables simply aligned by pure chance.
Exactly. The real shockwave in the cultivation world didn't come until the 1980s with an American mycologist named Ronald Ower.
Ower is famous in mycology circles.
He is. Ower developed a very specific protocol using nutrient-rich substrates to coax the underground sclerotia to produce fruiting bodies in a controlled indoor environment. He even secured patents for his process.
And the mycological world thought the code for the yellow morel had finally been cracked.
They celebrated. But the molecular phylogenetics we discussed earlier came back to haunt them.
When DNA sequencing became widely available decades later, the geneticists tested the strains originating from Ower's patented process.
And they discovered a monumental error.
It wasn't the right mushroom.
It wasn't. The mushroom Ower had successfully cultivated was not Morchella esculenta. It was an entirely different, genetically distinct, and significantly less finicky species.
A species that eventually became categorized as Morchella rufa-brunea.
Right. The true Morchella esculenta, the hyper-specialized yellow morel of the European and Himalayan hardwood forests, remained completely unconquered by science.
Because trying to cultivate it using standard fungal techniques is like trying to run a highly complex automated manufacturing plant without realizing you fired the entire logistics department.
It's a perfect way to put it.
You can provide the factory with all the raw materials, the carbon, the nitrogen, the water, but if the internal transport mechanisms, the specific temperature controls, and the waste management systems aren't perfectly calibrated, the assembly line simply shuts down.
You cannot separate the Morchella esculenta mushroom from its complex, invisible ecosystem. It demands the complete package.
And that realization is exactly what led to the massive modern breakthroughs we are seeing now, which began with large-scale outdoor field trials in China.
The Chinese cultivators recognized that indoor sterile cultivation simply wasn't working, so they moved the operations outdoors.
To closely mimic the natural diurnal temperature swings and the soil complexities?
Yes, and they innovated a really brilliant physical delivery mechanism for the nutrients, the exogenous nutrition bag.
This is a fascinating bit of agricultural engineering. How exactly does a plastic bag sitting on top of the soil feed a subterranean fungal network?
Well, the farmers prepare long outdoor beds of natural soil and sow the morel spawn. Then, they take specialized plastic bags filled with sterilized, highly concentrated carbon sources.
Usually wheat, cornmeal, or chaff, right?
Exactly. And they lay these bags directly on the surface of the soil bed. They slice a small opening in the bottom of the bag, right where it contacts the dirt.
And the fungus finds it.
Oh, it definitely finds it. The mycelial network growing beneath the surface senses this massive localized carbon source. It physically grows up out of the soil matrix, enters the slit in the bag, and begins rapidly secreting amylase enzymes to digest the complex carbohydrates.
So it accesses the food. But the goal isn't to grow mycelium inside a plastic bag. The goal is to produce sclerotia deep underground. How does the energy get from the surface back down into the soil?
Through a process called cytoplasmic streaming.
How does that work?
Fungal hyphae aren't entirely closed cells. They have pores in their septa, the internal dividing walls, that allow fluid and organelles to flow continuously throughout the entire network.
Like a plumbing system.
Exactly. The mycelium inside the bag digests the wheat, converts it to soluble sugars, and uses immense internal hydrostatic pressure to actively pump that liquid carbon back down through the vertical hyphae.
Deep into the soil bed.
Yes, where it fuels the aggregation and massive expansion of the subterranean sclerotia bunkers.
They are essentially force-feeding the network via an intravenous carbon drip, just to guarantee it has enough stored energy to trigger a massive fruiting event when that thermal window opens in the spring.
And initially the results were staggering. The yields were massive.
But it didn't last.
No. Within a few seasons, the entire system began to collapse. The Chinese farms slammed into a barrier known in agronomy as continuous cropping obstacles.
We see this in traditional agriculture all the time.
Very common.
If you plant corn in the exact same field year after year without rotating crops or resting the soil, your yields plummet because you are systematically depleting specific nutrients and inviting specialized pathogens.
It is the exact same principle, but amplified by the closed, hyperdense nature of the fungal beds.
What exactly was going wrong in the soil?
As the cultivated Morel mycelium grew to unnatural densities year after year in the exact same soil, it drastically altered the localized biochemistry. The soil pH dropped, becoming far too acidic for the Morchella Esculenta specialization.
But the sources say there was a much more critical issue, right? The network was suffocating in its own exhaust.
The ethylene gas. We are back to the ACC precursors building up in the soil matrix.
Right. Because they had artificially inflated the density of the fungal network with those exogenous nutrition bags, the respiration rates completely skyrocketed.
And the soil became super saturated with ACC.
And without the natural, balanced microbial ecosystem of a wild forest to act as a buffer, that ACC rapidly converted to ethylene gas.
Precisely. The toxic stress hormone accumulated, it arrested the hyphal growth, and the artificial beds effectively poisoned themselves. The yields crashed.
And this is where the sheer brilliance of modern, ecologically aware agronomy comes into play. Because to solve this continuous cropping obstacle, the scientists couldn't look at the mushroom in isolation anymore.
They had to look at its evolutionary partner.
They had to look at the secret agricultural phase we discussed earlier, this Pseudomonas putida bacteria.
Exactly. The researchers realized that the artificial beds were missing the logistics department. They were missing the biological scrubber.
So what do?
Recent trials have shown that if farmers intentionally inoculate the casing soil of these commercial morel beds with highly concentrated laboratory-cultivated strains of Pseudomonas putida, the entire system stabilizes.
They just artificially introduce the exact bacterial workforce the mushroom naturally evolved to farm.
Yes. The bacteria rapidly colonize the soil pores. They encounter the massive buildup of ACC exuded by the stressed fungal network, and they immediately begin metabolizing it using their ACC deaminase enzymes.
They scrub the precursor from the soil before it can convert to ethylene.
The toxic stress is lifted, the continuous cropping obstacles evaporate, and the artificial beds return to producing massive sustainable yields.
It is a profound shift, moving from simple monoculture farming to active, multi-species microbiome management.
It is a massive technological leap, and it's now being replicated and refined globally.
The papers actually highlight a particularly monumental, very recent breakthrough right in the heart of the Gucci's native territory.
Yes, driven by scientists at SKUST. That's the Shari Kashmir University of Agricultural Sciences and Technology in Kashmir.
A research team led by Professor Tariq Ahmad Sofi didn't just try to adapt Chinese methods, right? They set out to completely domesticate the wild Himalayan strains.
And their methodology highlights just how deeply you have to understand the ecology to actually succeed. They didn't just take a spore swab and put it in a petri dish.
They embarked on a massive, multi-year ecological survey, and the scale of the data collection is just mind-boggling. They trekked across the Pir Panjal range and surveyed over 1,000 distinct, highly isolated wild locations where the Gucci mushroom naturally occurred.
Think about the effort involved in that. And they weren't just looking for the mushrooms.
What were they doing at those sites?
They were utilizing advanced metagenomics to analyze the exact composition of the localized soil microbiome at every single site.
So they were mapping all the bacteria.
Bacteria, fungi, everything. They measured the granular fluctuations in soil pH, the exact mineral availability, the specific flora providing the leaf litter.
And they mapped the precise microclimatic data too, right? Like the exact degree of the diurnal temperature swings during the spring thaw.
They were building an incredibly high-resolution data model of that 100-million-year-old ecological niche.
And from those 1,000 wild locations, they isolated 10 highly promising genetic strains.
Then they brought them back to the lab.
Utilizing controlled polyhouses, these highly specialized agricultural greenhouses, they painstakingly recreated those exact, wildly specific Himalayan parameters.
They engineered the soil microbiome, matched the chilling requirements for the thermal window, and introduced the specific bacterial partners.
And by perfectly mirroring the invisible complexities of the wild ecosystem, they finally coaxed 3 of those pure, wild Himalayan strains to fruit reliably and predictably in captivity.
They completely cracked the code. They domesticated the 40,000 rupee prize.
It is a watershed moment in mycology. It takes a highly volatile, physically dangerous foraging economy and transforms it into the foundation for a stable, highly lucrative, and scalable agricultural industry for the local region.
And scientifically, it proves that the only viable path to mastering a complex organism is to deeply, intimately understand its ecological web.
Rather than trying to force it into a sterile, oversimplified agricultural box. It's an incredible narrative arc. Let's trace the path we've taken today, because the scale of what this organism does is kind of difficult to hold in your head all at once.
It is a lot to process.
We started with a biological entity that was wildly misidentified by the founders of taxonomy, lumped into a generic category, when its genetic truth reveals a 100-million-year divergence.
Clinging with intense evolutionary stubbornness to a highly specific alkaline hardwood niche.
And utilizing specialized aerodynamic honeycomb architecture to perfectly optimize its spore dispersal in the boundary layers of the forest floor.
We examined its remarkable metabolic plasticity, its ability to effortlessly shift from a mutualistic partner trading minerals with living trees to an aggressive saprotrophic miner dissolving dead matter.
Funneling those extracted nutrients into heavily armored, melanized underground sclerotia bunkers to survive catastrophic conditions.
And deliberately timing its awakening to an incredibly narrow, near-freezing thermal window to outcompete the entire soil microbiome.
Then we uncovered the staggering reality that this blind subterranean network actually operates as an active agriculturist,
manipulating the microphysics of soil water to build bacterial highways,
purposefully exuding carbon to rear localized populations of Pseudomonas putida, and eventually digesting them.
all as a highly evolved strategy to deploy biological scrubbers that consume toxic ACC and prevent the fungus from suffocating in its own ethylene exhaust.
We delved into its intricate chemical defenses, seeing how its evolutionary need to survive the toxic decay of plant matter resulted in the ability to neutralize furanocoumarins,
The very mechanism that holds the potential to solve the grapefruit juice interactions with human pharmacology.
We explored its payload of fuco-specific MEP5 lectins that actively downregulate inflammatory kinase pathways to protect the liver.
And of course, the critical, dangerous reality of its volatile hydrazine neurotoxins that demand thorough cooking to prevent severe neurological breakdown.
And finally, we saw how deciphering all of these invisible, highly calibrated mechanisms, understanding the carbon transport of exogenous nutrition bags, mitigating the autotoxicity of continuous cropping through bacterial inoculation, and those massive 1,000-site metagenomic surveys in the Himalayas, has allowed scientists to finally conquer this recalcitrant species.
Bringing the wildly lucrative, previously untamable Himalayan gold into the realm of precise modern agriculture.
It completely shatters the illusion that fungi are merely passive decomposers waiting for things to die.
They are active, aggressive, highly strategic engineers of their environment.
So, the next time you happen to step off the pavement and onto the damp soil of a forest or even just the mulch in your backyard, do not make the mistake of thinking you are just walking on dirt.
It is never just dirt.
You are standing on the roof of a staggeringly complex, chemically armed, biochemically intelligent infrastructure. You are walking over an organism that is actively sensing thermal shifts, synthesizing rocket fuel components for defense, and deliberately farming bacterial crops in microscopic water films.
It is an invisible intelligence that has been refining its engineering since the dinosaurs walked the Earth.
And that reality leaves us with a final, highly provocative thought for you to consider. If a relatively common fungus, something foragers have been tossing into baskets and sautéing in butter for centuries, has quietly evolved the capacity to construct transit infrastructure, synthesize highly targeted liver-protecting lectins, and systematically rear and harvest specific bacterial populations to scrub its own atmosphere.
What else is down there?
Exactly. What other forms of highly advanced multi-species agriculture are currently operating in the darkness beneath our feet, entirely undetected by modern science?
It's a huge unknown.
And as we continue to map those invisible subterranean partnerships, what unimaginable biochemical tools are waiting to be discovered that could completely rewrite the future of human medicine and global agriculture? Suddenly, the muddy, chaotic biology of the soil doesn't look like a mess at all. It looks like a master blueprint, and we are only just learning how to read the first page.